In contrast with vertebrate animals that have large muscles with many motor neurons to control their contraction, many invertebrate animal species are small, and since cells are generally the same size in both large and small animals have relatively fewer total body cells. Therefore, small arthropod animals such as insects must have small muscles that contain only a few muscle fibers that are innervated by only a few neurons. Despite these limitations, insect muscles are like vertebrate muscles in that they must be capable of making graded fine contractions for slow or delicate movements such as walking or manipulating objects. Yet, they must also be able to make strong total contractions that power rapid movements such as jumping, wing beats for flight, or as in this case, capturing and holding prey. Let us examine how arthropod muscles are innervated using the insect muscle as an example. Here is an insect muscle, along with the neurons that innervate the muscle fibers. Let us diagrammatically expand and separate the muscle fibers to better see the relationships between the muscle fibers and their controlling neurons. Because insects are small, they have only one or several fibers in any muscle, and only 1 to 3 neurons are used to control those muscle fibers. In this example, 3 muscle fibers make up this muscle unit with 3 controlling neurons. However, 3 fibers and 3 neurons does not mean that each fiber is controlled by 1 neuron. The neurons differ in their stimulatory actions on the muscle fibers and are known as fast, slow, or inhibitory neurons based on their effects on the fibers. Unlike vertebrate muscle fibers that are controlled by a single neuron, insect muscle fibers may have polyneuronal innervation. Polyneuronal innervation means that the contraction of a single insect muscle fiber may be controlled by 1 neuron, 2 neurons, or as many as 3 neurons. Not only may multiple neurons control a single fiber, but insect motor neurons have multiple neuronal end plates distributed over the entire surface of the muscle fiber. This is called multi-terminal innervation. Multi-terminal innervation occurs because action potentials are not propagated over the surface of insect muscle fibers, as they are for vertebrate muscle fibers. Insect end plates depolarize only the localized region of the muscle fiber near the end plate and cause this region to contract. Each motor nerve impulse releases a quantity of the amino acid L-glutamate that serves as the stimulatory neurotransmitter chemical for arthropod muscles. Here we see a close-up of a neuron motor end plate showing glutamate-responsive sodium channels on the muscle fiber surface. Repeated nerve impulses cause increasing release of glutamate, resulting in additive electrical depolarizations and increased contraction of the arthropod muscle fiber. The increasing degree of depolarization and contraction in response to multiple repetitive motor nerve impulses is termed facilitation or temporal summation. Let us now see how 3 neurons can account for the range of both rapid and fine-controlled graded movements that insects can make. Three types of neurons innervate arthropod muscle fibers. All insect muscle fibers are innervated by fast motor neurons. Here we see a fast motor neuron with 3 branches. Each muscle fiber is innervated by a branch of the fast neuron. Fast motor neurons produce rapid total contraction of the muscle fiber. Some fibers of insect muscles are also innervated by slow motor neurons. Slow neurons control the partial graded contractions of the fibers. The differences in action by fast and slow motor neurons are based on the amount of glutamate neurotransmitter that is released by each type of neuron and the amount of summation effects. Fast neurons release large amounts of glutamate, resulting in intense depolarizations with little facilitation or summation as compared to slow motor neurons. Slow motor neurons must fire repeatedly to cause a given muscle fiber to attain the levels of depolarization and contraction that are comparable to those for a fast motor neuron, and facilitation and summation are common. Note that since only one muscle fiber in our muscle unit has slow innervation, not only is the contraction rate of the entire muscle slow and graded in response to stimulation by the slow neuron, but the overall maximum contraction of the entire muscle is only about 1/3 of that observed for stimulation by the fast neuron. Some muscle fibers are also innervated by inhibitory neurons that block the stimulatory effects of both the fast and the slow neurons. Inhibitory neurons release gamma-aminobutyric acid as their neurotransmitter chemical. Gamma-aminobutyric acid hyperpolarizes the fiber membrane to neutralize the depolarizing action of glutamate released by the stimulatory neurons. Let us now see how inhibitory neurons interact with stimulatory neurons for adding additional controls to insect muscle contraction. In our first example, the fast neuron stimulates all three muscle fibers to provide a maximum contraction. Next, firing the inhibitory neurons hyperpolarizes muscle fibers 2 and 3 at the same time as they are depolarized by the fast neurons. The inhibitory hyperpolarizations block the stimulatory depolarizations to prevent muscle fibers 2 and 3 from responding. Note that only muscle fiber 1 undergoes depolarization and contraction, so that total muscle shortening can only be 1/3 of the shortening which is seen when all three muscle fibers are stimulated. In the second case, we see the graded contraction of the muscle by the slow neuron. Note that the contraction is only 1/3 shortening of the entire muscle, since only muscle fiber 2 has the slow innervation. When a slow neuron stimulus is combined with stimulus by the inhibitory neuron, the slow neuron stimulation is blocked completely, and muscle fiber 2 is completely suppressed, so there is no muscle contraction. Finally, in the last example, both the fast and slow neurons fire simultaneously with the inhibitory neurons. Again, the muscle shortens by only 1/3, since only muscle fiber 1 is free to respond to its fast neuron. Polyneuronal innervation of single muscle fibers and multi-terminal end plates over the surface of the muscle fiber are adaptations to small size that allow small arthropod animals such as insects to compensate for having fewer muscle fibers than are found in the larger vertebrate muscles. In this way, arthropods minimize the number of neurons that are needed to innervate the muscles, while preserving the range of graded and fast contractions that are essential for normal movements and activity.